Okay, today we're going to talk a little bit about doing science and the scientific method. We've talked a lot already about measurement and how that's used, so now we're going to tie in the scientific method that when we actually do labs and we do activities, we have to be able to use different steps to get our measurements, and those measurements, again, always have to be accurate and precise, have the correct number of significant figures. So here are the what we call the steps of the scientific method. But if you notice here, there's no one scientific method with an X number of steps. There's common processes that we use, but we don't always do these things in order. So questioning and observing, gathering data, analyzing the data, using that to draw conclusions, and communicating your findings. These 5 different things aren't always done in a certain order. It depends on what we're trying to do. You can also see here that there's a lot of different ways that we gather data. It can be anything from reading literature to doing a survey to taking observations of over a long period of time, or just doing an experiment like we typically do here in chemistry class. So what we say is that science is loopy. There's no one way. Okay, so we could take observations. We might do that first and then go to a question that comes up from seeing an observation. Then we make a hypothesis, or sometimes we would go directly to gathering data. If something's happening right that second and you have to have that data right then and there, then you need to be able to do that right away. So what this is showing you here is that loopy means you might not always start and go in the same direction. Here we might have a product, and then we have to go back and make observations and start again and go back into this loop. So sometimes we're going to go through the process, have to go back up and redo steps, and this just shows you all those different pathways that are possible. So again, remember that there is not just one way to do the scientific method. 2 main types of experiments that we're going to be doing: investigating relationships or effects. So how does one thing affect the other? For example, volume affecting pressure, or reaction rate changing with changing temperature. We'll do some of those in class. We'll also do some of these where you're only going to determine a specific value and you're just going to figure out a number. Okay, so we deal with those 2 different types of experiments very different. You have to understand your variables. We did a lot with this last year in biology. We have an independent variable. You control that, or I control that. I remember there's an I here, so I control that variable, and then the dependent variable is what you measure. So if we read this example, how does reaction rate change with temperature? What am I going to control? Well, I can't control how fast the reaction goes, but I can change the temperature, and I can control how hot the water is for that experiment. So what then am I going to measure? I'm going to measure how fast the reaction takes place, or the reaction rate. Okay, so that's how we can look at an experiment to determine which is our independent and dependent variable. And we need to be able to do this to make a graph correctly. Okay, so we'll be using that later on to make graphs. Independent and dependent variables go on the different axes. Another example here, what is the concentration of that salt solution? Well, here I'm not really controlling anything. I'm just figuring out a value. So if I'm figuring out a quantity or a value, there's not going to be an independent or a dependent variable. I'm just going to try to find an answer that's going to be numerical. Now, when we do an experiment, we know that we can only change one variable. We have to hold all our other variables constant. Okay. So the independent variable is what's going to be changing. The dependent is what we're going to measure. The rest have to be constant. So if we're talking about this experiment here, where how does the reaction rate change with temperature? If we're using water, we have to use exactly the same amount of water every trial. We have to keep that constant. Okay, that's something that we cannot change. So as we go down here, we also have to keep our concentration the same. So if we're using salt solution, it has to be the same concentration every single time. The other is the method of how we determine the rate. So if I'm going to measure the time from when I first combine the reactants till I see no more bubbles, then I have to do that exactly the same every time I collect data. Okay. Now, another thing we do in scientific method is hypothesis. We attempt to explain why something happens. So a prediction, remember, it's just a prediction. It only predicts. The hypothesis attempts to explain why that happens. And this is why we have to have that because statement in there. Okay. So how does surface area affect reaction rate? A prediction would just say that reaction rate is going to increase as surface area increases. A hypothesis says if surface area increases, then the reaction rate will increase because more particles have a successful collision, okay, at the same time. So there's more contact. So for a hypothesis, we have to have that because statement because that's the explanation. Okay. So what is the concentration of that salt solution? We're going back to that. This again was one where we're just determining a quantity or a value, numerical value. We cannot write a hypothesis for that. So remember, anytime you do an experiment where you're figuring out just numerical data, you're not going to have a hypothesis or an independent or dependent variable. Now here we've already talked about this a little bit, precision and accuracy, but some of you missed this on the test, so this is a great review. Precision is being consistent, accurate is being correct. Okay, so again, looking here, our correct values in the middle, this would be precision because we're all grouped together. They're being consistent. Here we're correct and we are consistent. So whoops, this would be accuracy and precision, and this one would be neither. Okay, and again, we already looked at this earlier this year, so I'm going to kind of go through the next couple slides that talk about that. Okay, again, you can't really be precise if you're not accurate. You can't be accurate, excuse me, if you're not precise. So if you're accurate, you're going to have all right answers, and they're all going to be grouped together there. Again, we're going to have some experimental error. We talked about that too. That, you know, if you're transferring something from one container to another, you're going to lose a little bit, most likely, and so that's going to lead to some error. You cannot blame error on yourself or on the equipment, though. Remember that. Another step in the scientific method is drawing conclusions. So what we do is look at all the evidence from the data, and we try to draw a logical conclusion. Okay, sometimes they support the hypothesis, sometimes they don't. It just depends. And again, we did a lot with this in biology last year. You guys did it with the growing pea or soybeans, and you made a hypothesis if you changed the water, if you changed the fertilizer, what was going to happen. Some of you supported your hypotheses, some of you didn't. Sometimes this will lead to a new investigation, and sometimes it will prove that we're wrong for something, and that's perfectly fine too. But drawing conclusions is another step in the scientific method. Now, theory versus law, again, very similar, but the theory is why something occurs. The law describes what happens. A theory cannot become a law. So those are two real important things to understand. A law, a lot of times, is mathematical. It's an equation that we can use to figure something out. So here's an example here. The kinetic molecular theory explains why something happens, that when we decrease volume, the number of frequencies are going to increase, so the pressure increases. Where Boyle's law tells what happens. If we have a pressure and a volume and that changes, we're going to get a different pressure and a volume. And again, you can figure that out mathematically. The last step here that we're going to talk about, again, it doesn't have to be the last step, but it's the last one we're going to talk about, is communicating results. When we're done, we have to be able to share what we found out. So a lot of times in your guys' lab, you have to talk to another group and see if they agree or disagree with you. So we're adding to the pool of knowledge. Sometimes in scientists, they're going to do presentations or posters at a conference, and I've done this. Mr. Nallen and I went out to Washington, D.C. last year and presented at a conference doing a poster presentation. Sometimes they write articles. Sometimes it's online collaboration and discussions, like through blogs and things like that. And sometimes it's collaborating with another group and working together to figure out if we can get the same answers. So that would be the last step that we're going to talk about in the scientific method.